Welcome everyone to this week's edition of the Extracellular Vesicle Club. I'm really happy today to be able to introduce Marc Poirot. He is with INSERM, and he's also at the Cancer Research Center in Toulouse in France. And recently his group published this very nice paper on how EVs can actually be immunogenic in cancer. And this was a publication in the Journal of Extracellular Vesicles, which is ISEV's first and flagship journal. So, Marc, thank you so much for joining us today. And I'd like to invite you to go ahead and share this exciting work with the audience.
Thank you, Kenneth. It's very nice to be here with you, and thank you for the invitation to present our work. So I will talk about these properties of a molecule of the cell, adenosine A, that we found to be very active to induce modification and the secretion of secretory vesicles. And these vesicles became immunogenic, and we found that actually these exosomes induce an immunogenic response against the tumors and take part of the mechanism of action of these molecules, adenosine A. So, okay, so we used to consider that secretory vesicles include exosomes that are produced from multivesicular bodies, and also microvesicles. They contribute to these vesicles can be a lot, so more than around 500 billion vesicles potentially released by million cells per day, which is a huge amount potentially. And we used to separate the microvesicles from exosomes because the way they are produced are quite different. And in lipids are a very important component of these mechanism. And it was reported that bis-monoacyl glycerophosphate, a very peculiar lipid, is involved and associated with not only multivesicular bodies, but also with exosomes. And it's important this lipid contribute to the production of these vesicles. And usually there are, what we found in the cellular medium is a mixture of these two kinds of vesicles, exosome in one hand, and that are produced from intraluminal vesicles, and extracellular vesicles that are co-purified with them when we are proceeding to ultracentrifugation, purification of these vesicles. And it has been reported since years that they could contribute they could promote cancer development through different kinds of mechanisms. For example, they can squelch drugs and limit their efficacy towards tumor cells. They can also produce an immunosuppressive microenvironment with several mechanisms, and they could favor the production of a premetastatic niche. So all these make these vesicles of very interesting of interest. So now adenosine A is a compound we discovered in the team. This is a new kind of steroids. It's a steroidal alkaloid, which is a chimer between cholesterol and histamine. And this exemplify or highlight the fact that we have discovered a new metabolic branch on the cholesterol pathway. And I'm very proud because I was recipient this year of the Stroppmer Medal that recognized the importance of this discovery. And I have already made a webinar through lipid maps that can be accessed on the web, and will tell you about this kind of discovery. We found that DDA is present on a lot of different tissue. We have not explored all tissue, but we found it in the liver, which is one of the major tissue containing DDA. It's present in the breast also, and several other tissue. And what we found of interest is that when we checked the production of DDA in normal breast compared to breast cancer, we found it decreased drastically. And we found another steroid that is different, but display tumor promoter properties, this one, which is oncosterone. And that's highlight a key metabolic deregulation in this cancer. And what was interesting also is that DDA is absent from most cancer cells of different tissue origin. Now, if you add DDA to cancer cells, you can induce first cell differentiation and growth arrest, and then for higher concentration, this will give a lethal autophagy. Then cells will die from autophagy. We have also defined the mechanism. DDA acts as a ligand and modulator of elixir, but instead of activating canonical genes known to be under control of these nuclear receptors, we found that it can activate different expression of genes involved in the production of differentiation markers, lipid biosynthesis enzyme, and this is required to induce cell differentiation. Otherwise, we found it induced the expression at the transcriptional level of autophagic proteins, lysosomal proteins, and also several lipid biosynthesis enzymes involved in the production of autophagosomes and lysosomes. And this led to the induction of lethal autophagy. And what was also interesting is that endogenous or synthetic agonists of elixir beta inhibit or do not produce these effects at all and inhibit the effect of DDA. So when we check first the impact of DDA on tumor implanted in immunocompetent mice, this is the example of a model of melanoma cell, B16F10 cells, which is well known. You can see here by comparison with dacarbazine, which was a cytotoxic use at that time, that DDA was very efficient at low concentration to control the development of tumors. And most importantly, this improved mice survival drastically. We found that at that concentration of DDA, we induce tumor cell differentiation. This is Fontana-Masson staining, which highlight stain melanin, and melanogenesis is associated with melanoma redifferentiation. And interestingly, we found also that we got a strong infiltration of T lymphocyte and dendritic cells. And what was interesting at that moment is that we found that the same experiment done on the nude mice does not give that results, suggesting that at that concentration, DDA was inefficient on nude mice to control the growth of B16F10 cells. And that suggested that maybe this immune infiltration was related to the anti-cancer action of DDA at low doses. So we found that, for example, DDA induced B16F10 redifferentiation with a strong accumulation of cells in G0, G1 cells, and also a morphological modification characteristics of of melanocytes. Same was observed with human melanoma cell lines, such as SKML28. You can see here a strong morphological modification. In each case, we activate the production and the secretion of melanin. And we asked the question whether or not DDA could modify vesicle secretion and function by differentiating tumor cells. And this is electron microscopy of cells here, control, compared to cells treated with DDA. And you can see that we have a huge production of multivesicular bodies, and you can see at the surface of cell the production of vesicles that can be of two origins. First, we have exosomes from multivesicular bodies, and we got also secretory extracellular vesicles. We found that I was talking about the BMP at the beginning of my talk, and we found that DDA stimulates drastically the production of BMP within cells. And this effect was interesting because we tested another elixir ligand, and we found that it decreased the basal level of BMP compared to control and inhibit the effect of DDA, suggesting on BMP production, suggesting that this effect was elixir dependent or elixir mediated. So we went on the characterization of these vesicles, and first we found that the stimulation of SAV production through DDA stimulation was concentration dependent, which is suggesting that this effect was mediated by a receptor. And we characterized these vesicles and found the expressed tetraspanins, which is what we expected to obtain, and found that by FACS analysis, because there's a good antibody can be useful for FACS analysis for BMP, we found that DDA stimulated the production of BMP, but also we got a modification of the phenotype. Not that much an effect on ALIX or TCS 101, but a decrease in flotilin and a stimulation of tyrosinase and annexin A1 and also SC3 2. Now, we found also when looking at cells that BMP was present in BMP. This is immunogold electron microscopy analysis, and these black dots are BMP labeled by the antibody. And we found also that LC3 was present on these MVB, suggesting that not only LC3 could be important for exosome, for autophagosome biogenesis, but also could be involved in the production of MVB intraluminal vesicles. And we found also that DDA stimulate the production of calreticulin. So next, we tested these exosomes in tumors implanted in mice, and what we found is that 2 injections induce a strong control of tumor growth, while constitutively produced ACV by cells have no significant impact on the development of tumors. And that was very important because it was the first time that it was shown that a vesicle like that could be modified strongly and induce control of tumor growth when injected in mice. So what about the mechanism? We found that on B16F10 that an agonist of elixir, the 22R hydroxycholesterol, do not induce the production of ACV like DDA. And SKML28, we were able to transfect these cells with single hairpin construct against Elixir beta. And we found that while DDA stimulate the expression, the production of ACV, this was abolished when Elixir beta was knocked down. We found that on another kind of cells. These are mouse breast cancer cells, the EO771, and we succeed in knocking down Elixir beta in these cells. And again, we found that DDA stimulate the production of ACV in these cells, but that Elixir is required to induce this effect. And finally, we use mouse embryonic fibroblasts from Elixir beta cow knockout mouse. And again, we found that while on the wild type MEF, DDA stimulates the production of secretory vesicles, we found that knockout MEF, DDA was unable to do that, and that we were below the background, suggesting that Elixir beta could play a very important role in the production of these ACV. On SKML28, we found that the absence or the presence of Elixir beta do not impact on the production, on the expression of tetraspanin on these cells. But we found that there was a strong decrease in BMP on this ACV. And we found also that the phenotype induced by DDA was drastically modified. This includes melanocytic differentiation antigens, also proteins involved in the control of MVB secretion, proteins involved in immunogenic cell death, and some effect on escort proteins. And what we have done is that we have tested whether what could be the impact of DDA ACV on the immature dendritic cells, and compare the effect on DDA ACV obtained from SH Elixir beta of Elixir beta knockout cells. And we found actually that in the first case, we got mature DC that express a number of markers of activation of dendritic cells, such as HLA-DR, CD86, CD54, CD40, CD80, CD83. And this effect was not observed when we used ACV, DDA ACV from cells knocked down for Elixir beta. So that shows that Elixir beta is really required to the production of ACV, able to induce the differentiation of DC into mature DC. And when we check what the impact of these cells on naive T cells, we found that on the first case, we got a strong Th1 polarization with the production of IL-12, IL-6, TNF-alpha, interferon gamma, IL-13, and IL-5. And this effect was not seen with immature DC produced with DDA ACV from Elixir beta knockdown cells. But together, we found that tumor cells treated with DDA get differentiated in an Elixir beta dependent manner, and these cells produce ACV enriched in a number of marker and immunogenic proteins, markers such as BMP, but also tyrosinase, melanoma and a number of proteins. These ACV can induce the differentiation of immature DC into mature DC and induce Th1 polarization. And this is probably related to the induction by this ACV of the anti-tumor effect we found on the B16F10 tumor cells implanted in mice. So we found that it is pharmacologically possible to change the phenotype of ACV and to transform them into immunoactive one. We found that DDA, which is a cholesterol metabolite, can do that. These data illustrate the fact that XR beta can play a new role as an anti-cancer target, but it depends strongly on the chemical structure of the ligand we will use. And this also highlights a new role for XR beta in the biosynthesis of BMP and other lipids. So we can ask also, because DDA is not, it's a molecule of the self, we produce it in a number of tissues. So these compounds can contribute not only on the epithelial function of normal cells, but also be involved in the immune surveillance of healthy tissue. And we are looking at why we found that DDA is downregulated during the occurrence of several cancers, in particular in breast cancer. And since we found a family of compounds related to DDA, we are looking at the effect or the impact of these new steroid metabolites on ACV formation and phenotype. So I want to thank in particular Sandrine, who is the co-head of the team, Michel, who initiated the project when he came in the lab, and Philippe, Julio, Celia, and Kevin, who contribute to the development of this project, and also all the sponsors of our team. So I thank you for your attention. I'll be happy to answer your question if I can.
Well, thanks very much, Marc. This is very exciting work, and I have a few questions. But before we start, let me just remind everybody that if you would like to ask a question or make a comment, please just put that in the chat box, and then we will get to you in order. And I am going to allow unmuting and screen sharing now, camera sharing if you want to. So Marc, let me start out. You had administered in your in vivo work, you had administered the DDA EVs, so the ones that had been produced under the influence of DDA, and you had injected those intradermally, I believe. Is that right?
Yes.
So what about route of administration? Do you think that that's the only way that you could do it, or is the goal there to get as close to the tumor as possible? Because I think you actually were doing it on the other flank.
Yeah, we have done both on the other flank or around the tumors, and we found similar results.
Okay. So, envisioning a future trial with human patients, how do you think you would be administering these EVs there?
Actually, this is not a question I asked, because what we wanted to look at mainly was the mechanism. We wanted to study exhaustively the activity of donor A. So we are working with a company who is developing these compounds for clinical application, but this is their job, not us. So
sure. Yeah. Understood. Yeah. So what about administering the dendrogenin A directly? I mean, as you mentioned, this is a metabolite. I mean, would it be helpful to do that, to administer it directly?
Actually, yes, we observed a very strong, very interesting effect with dendrogenin A, and something we have not yet done, but that would be also very interesting, is to test a combination with exosomes, DDA exosomes, and DDA, because we found that the effects were not exactly similar. So we suppose that they might be different aspects. We know that DDA as an elixir ligand may affect also the immune system, because elixir is well known to control the immune response by itself. But I think the fact might be more complicated than expected. So there are plenty of experiments that we need to perform in order to better understand what would be the advantage to use one or the other, or a mixture of both.
Yeah, very good. Now, you mentioned in your conclusions that you're also interested in understanding the DDA dynamics, I guess, for lack of a better term, in cancer. So how do you plan to go about that, to understand how this particular metabolite is being regulated, is being used, is being processed, I guess, in cancer?
Actually, what we have done is we have identified the enzyme involved in the biosynthesis of DDA, and studied its expression during carcinogenesis, and found it was downregulated. So we found that, and it probably due to a control of expression through epigenetic mechanism. So right now we have performed analysis of a cohort of patients with 300 patients with different kind of subtype of breast cancer, and this is something we observed. And so that's the first thing. So the decrease in DDA is due to the decrease in the DDA synthase. And second, we found also that I was talking about the oncosterone. So oncosterone is another oxysterol that can inhibit the activity of the DDA synthase directly, and also that induce an effect on the immune system. This is something we have not yet published, but and so that's a conjunction of effects that could impact on the physiology of the breast, and that could be related to the dynamic of the cholesterol metabolism. And so this will be the next step we will publish. One of the study will be published this summer. Probably we will submit it at the beginning of the summer, and the other maybe a bit later. But this will help to better understand the dynamic of cholesterol metabolism and its implication on the development of cancer.
Very good, very good. So I got a private question here that was sent. Okay, this is good. When we hear about cholesterol, we naturally think about nutrition too. Is there a role for dietary interventions that might be suggested by your results?
Sure, that's a very good point, and this is something we're studying. Actually, we have several programs on that topic. And also, we haven't published that right now, but this is a study in preparation in which we show that, yes, cholesterol from diet and transformed cholesterol in particular may have a very strong impact and favor the cholesterol degradation in normal tissue, and favor carcinogenic processes.
That's fascinating. I think we don't know very much at all about diet and EVs. So that's very interesting work.
Yes, very interesting. And you will see that in the breast, well, this is something that I can tell. The DDA synthase is expressed on the epithelial cells from lactating duct. those cells that are at the origin of 70% of breast cancer. But I think that EVs that are produced in the milk could be related to those that are induced by DDA and develop and display also anti-tumor properties. So that could be also a link between milk and the functionality of some EVs that were already reported when you take them from milk and breast cancer. So I think that would be also very interesting a field to look at.
Very good. Well, we're going to look forward to learning more. So Michael Grainer, you have a question here. Please go ahead.
So yeah, I was curious. It looks as if from the use of the DDA stimulated EVs, so you DDA stimulate the cells, they produce EVs, and then you can inject those EVs, which you did essentially, apparently dermally, which one presumes that targets the Langerhans dendritic cells under the skin there. But then what I was wondering is, to follow this up, I mean, you can continue to vaccinate, but it seems as if to keep generating EVs from the tumor itself, as you pointed out, by either the DDA treatment of the tumors, however that works, or by enforcing DDA synthase stimulation, it seems like that would be a pretty interesting idea, because as the tumor evolves, the antigens are going to evolve. So rather than trying to continuously keep up with that with the vaccine, you essentially make the tumor make its own vaccine. Yeah. But you might need a prime boost sort of setting to do that, which is why I think the dendritic cell or the EVs used are interesting.
That's a very, very good point, actually. And we have unpublished data related to that. Yes, that's really, really interesting. We already found that there are several compounds in the diet that are known to have chemopreventive properties on breast cancer that actively that stimulated the expression of the enzymes involved in DDA biosynthesis. So that's something that is realistic from yes, to activate this process might be very protective. We know also that unfortunately this enzymes is downregulated during aging, so that's also a factor that favors the development of breast cancer in adult people, and also to be able to stimulate or to maintain a level of expression that could produce DDA would be also very helpful to protect people for long term.
Great suggestions. Thanks, Michael. Is there anybody else who would like to ask a question of our speaker today? If not, I'd like to thank you very much, Marc, for sharing this work with us and for your responses to the questions. And we certainly wish you the very best as you continue, unveiling the secrets here. And we'll look forward to seeing more coming out of your group. So. And thanks everybody for joining today. We appreciate it, and we hope to see you again on a future edition of the EV Club.